across these different species: human, monkey, mouse, rat, xenopus, and chicken. The same goes to other amino residues. If particular amino residues are completely conserved in this multiple sequence alignment, meaning that the score value is highest. If less conserved, as indicated by double dot and single dot, the score value for these amino residues in MSA in multiple sequence alignment is lower than that of star symbol. Okay, so basically, the higher score value from this MSA indicating the more conserved proteins in different organisms. This is what we can obtain from multiple sequence alignment using cluster W or other bioinformatic tools. Okay, moving on to other bioinformatic tools, we can perform analysis not limited to protein sequence analysis. I mean identifying the conservation similarity, also not limited to doing multiple sequence alignment, but we can also perform other type of analysis. For example, we can identify protein domains or functional motifs using, for example, ProSite database, ProSite software. Similarly, we need to paste our protein sequence in the query box, click the button, and wait for the result. If we want to carry out 3D prediction of protein, we can use Swiss model software. I know the current one is called yeah. Any idea about I think the most popular tool that we can use to predict protein structure? Any idea about this? This one is very popular because the Nobel laureate candidate has used this tool to predict protein structure and solve a very significant biological problem. That bioinformatic tool is used to predict, to estimate, uh, to calculate the overall structure of protein biomolecule. Ada siapa tahu tak apa nama tool tersebut? Ada respon nampaknya. Okay, it is called AlphaFold. AlphaFold, which is also important in predicting 3D protein structure accurately. It is considered as a more advanced tool to predict protein structure apart from Swiss model. So in XPC server, we have Swiss model, and to initiate this prediction process, we need to click several buttons. For example, start modeling button, then we need to paste our query protein sequence. Okay, Swiss model software for 3D prediction of protein structure. Okay, now we want to see how Uh, the previously mentioned public protein databases and software, uh, how they are related to mass spectrometry analysis. And previously, I mentioned that we perform MS analysis. We obtain the spectra, and the spectra are used in a special software to identify individual proteins. So from MS analysis, we can obtain PMF, peptide mass fingerprinting. So PMF actually refers to a set of mass spectra obtained from MS analysis. Different proteins, different proteins would show different PMF, would show different peptide mass fingerprinting. So based on PMF, we can identify individual proteins using a special software. For example, mascot. The most common one, probably it is about, not sure, could be 50-year old, I think. Another software that can use to interpret PMF from MS analysis is MaxQuant. So all can be used for MS analysis and proteomic analysis. So mascot analysis is actually linked to Swiss spot database. PMF obtained from MS can be used in mascot software, and mascot software will interpret for us, will interpret PMF so that we can identify the individual proteins from biological sample. For example, mass spectra obtained from MS, they are interpreted and they will show two types of ion as we discussed before. They are B ions and Y ion. B ion is produced from N terminal, usually on the left side. Y ion is produced from C terminal region on the right side. Okay. So determination of amino acid sequence happen in two directions, two opposite directions. From left to right for B ion, from right to left for Y ion. So this mascot software or any specific tools will interpret this sequence and compare their opposite direction so that the most accurate sequence can be obtained. Okay. For example, here we can see the sequence of B1 until B19 is identical to Y1 until Y19. It is about the double checking process to ensure that we can get the most accurate sequence of amino acid. Okay, mascot available in XPC server. It is used for protein identification. Okay, a front page for mascot software. So we need to key in the relevant input here. If we want to change the parameters such as peptide charge, peptide tolerance, post-translational modifications is up to us and also depends on our objective of experiment.